Our next topic is from plant anatomy. We study a short topic, periderm. The periderm is a protective tissue of secondary origin which formation by lateral meristem developing in extrastellar region. It replaces the epidermis when it destroys at the axis, increase its girth by extensive secondary growth. There is much variation in the age if the shoots when the formation of the periderm starts. Structure and development pattern of periderm. The periderm includes three parts we divided in three regions, the phallogen and its two derivatives phallum and phallodrome. We first study phallogen. The phallogen or cork cambium is a lateral meristem which forms cork or phallum towards the outer side and secondary cortex of phallodrome towards the inner side. Phallogen originates from relatively mature but living cells. Phallogen cells divide both in periclinian and anticlinian lines. After each division, one of the daughter cells remains meristematic and the other daughter cell gives rise to phallum. If it happens to lie towards the periphery of the stem, or phallodrome, if it happens to lie towards the center of the stem, it's known as phallodrome. In most of the woody plants, with the continuation of the process of secondary growth, the original phallogen is replaced by successively more deep-seated phallogens. In all stems, the later formed periderm layers are found in secondary phloem. Each of these phallogens functions normally and produced cork and phallodrome. As a result, pockets of dead cortical cells and phloem tissue becomes isolated between these periderms. These tissue make up the rhytidome. If the phallogen forms complete cylinder around the stem, the activity results in the development of a ring bark. On the other hand, if the phallogen is formed in orcs, this type of scale bark is formed. Next is phallum. The cells of the phallums or cork are compact or dead at maturity. They are characterized by suberin deposits on its walls. Electron microscope studies have revealed that the suberin lamella is layered. Sometimes non-suberized cells, also known as phalloids, also occur in the cork. For example, in betula, alternating layers of suberized and non-suberized cells are present. Consequently, the cork peel off like sheet of paper. Phallum being thick-walled prevents the inner tissues of the stem from drying out. And the part C is phallodrome. The phallodrome or secondary cortex is made of living cells with non-suberized cellulose walls which have simple pits. They contain protoplast like the parenchyma of the primary cortex. They sometimes contain chloroplast and function in photosynthesis and food storage also participate. The regulation of development of cork development process. Several environmental and endogenous conditions induce cork formation. Environmental factors such as submergence in water, direct strong sunlight, mechanical and biotic sounding are the factors to increase the production of gases phytohormone ethylene. It is therefore proposed that the ethylene is the major activator for the phallogen initiation activity. Since the cork layer formed is almost impermeable to gases, hence as a cork layer is formed, the inner cell layers are exposed to increasing level of ethylene and more cork layer is induced. In many plant species, there is a gap in phallogen initiation around Hudson and nodal region or a cork-free region in stems and branches beneath buds and major veins of leaves. Therefore, it is proposed that the basilar polar oxygen transport inhibits core formation in this region. Now, another term we study is rhytidome. Bark comprise all the tissue outside the vascular cambium in stem and roots. The majority of the bark of woody plants develop from three meristems: the vascular cambium that gives rise to the secondary phloem, the phallogen or cork cambium that produced cork and dilation, meristem that produce parenchyma cells to prevent cracking. Bark is an incisive term for all tissues outside the vascular cambium. Thus, it includes primary and secondary phloem cortex. It also includes the tissue formed by the dilation growth. Structurally, bark can be differentiated into two regions, the inner bark and the outer bark. The inner bark includes the innermost area of the periderm. It is alive and some of its cells may be differentiated and become meristematic. The outer bark includes the dead tissue on its surface of the stem along with parts of innermost periderm. When the formation of the periderm, the tissue external to the gets cut off from the inner tissues. Consequently, the water supply to outer bark stops and eventually it dies and becomes cracked, forming hard crust on the surface of stem. The tissue becomes more and thicker, and additional cork layers cut off by underlying living tissue are dead. The thick outer bark along with the layer of tissue it is enclosed, it's called the rhytidome. Commonly, all the tissue that is peeled off is bark and the name rhytidome is only for the outer bark. When the subsequent periderms develop in the form of overlapping scale of cells, outer layer are slowed accordingly. This type of bark is known as ring bark or intermediate condition found in eucalyptus. The last term we study in periderm, the lenticels. In the periderm of most plant cells, small areas of loosely arranged cells are present. These areas are well known as lenticels. Lenticels are usually found below a stomata. A group of stomata at such places, the phallogen is divided in different planes to form a mass of loosely arranged, rounded, unsuberized cells with many intercellular spaces. The resulting tissue is known as complementary of filling tissue. The pressure exerted by the formation of complementary cells ruptures the epidermis, thus exposing the mass of complementary tissue. Lenticels help in exchange of gases between the exterior and interior of the stem. That's all about periderm. Thanks, students.